A warm welcome, dear friends, back to our quest in understanding the tools for molecular cloning. In the last discussion, we had insight about the various enzymes which were needed for this process. Today, I would like to enlighten on the plasmids as a vectors for the tools for molecular cloning. Plasmids, these are one of the first requisites, you can say, the basic necessity for molecular cloning. So what are these plasmids, what types are there, how they evolved, what is their history? This will form the part of today's discussion. Now, as you know very well, molecular cloning is basically a construction of a recombinant DNA molecule. The gene of interest which we isolate, it has to be multiplied first of all, so that you get multiple copies of that gene of interest. Second thing is that it should retain its integrity, that is, it should not be degraded in a host. The third aspect is that it should have sustainability till the production or the expression of the genes for which it is meant for. So what you need is a plasmid as a vector. The gene of interest will be annealed into this with ligation, of course, and you get a recombinant DNA molecule or a chimera, which is nothing but a clone, which is a molecular clone when it will be cloned into multiple copies. This recombinant DNA technology, as I have been continuously telling you, is synonymous with the words genetic engineering, gene cloning, molecular cloning, so on and so forth. So whatever the terminologies which you hear, don't get confused. These are one and the same. We are moving towards understanding a topic or understanding a procedure where we are trying to engineer the genes. Taking the gene of interest, gene of a human interest, when it is said gene of interest, it is the gene of human interest. By that way, I think this technology goes against nature from one perspective, but from another perspective, with growing demands, growing population, we need more and more resources. It may be food, it may be the medical products which are needed, so on and so forth. There are n number of applications. So all this has been clubbed into one concept in your curriculum here, and that is called as molecular cloning. So what is this recombinant DNA technology? Just for your recap once again, recombinant DNA is the formation of a new species or a new DNA where these are coming from two different organisms. Now here the boundaries of eukaryotes and prokaryotes, these are dissolved. A eukaryotic gene can be expressed in a prokaryotic system. That is the beauty of recombinant DNA technology. And these recombinant DNA molecules, they can be made with recombinant DNA technology, which is known as molecular cloning. That is what we are going to explore. So the basic procedure is to cut the DNA of the donor organism, that is the source organism, into pieces and insert one of this fragment, which is that one, that is the gene of interest. Now believe me, having said these lines, it is very simple for me to say and for you to listen, but this gene of interest or one of this fragment is just like finding a pin in the haystack. That is, it is finding a needle in the haystack, that one gene of interest. But once that is isolated, you have to clone it into a plasmid and then proceed further with the aspects of molecular cloning. Now genetic engineering can be done in animals, in plants, in bacteria. There are various uh, windows open for this technology. There are newer vistas which has been explored nowadays. All this I have explained in the very first lecture of this unit, that is what is the basic of molecular cloning. You can watch that video. So the history, where did it all start? Where did the recombinant DNA technology get incepted in whose brains, and what is the origin of that? It was started in 1970. Hamilton Smith at Johns Hopkins Medical School, he isolated the first restriction enzyme which cut DNA at a very specific nucleotide sequence, and within next few years many more such type of restriction endonucleases were discovered. Today, as I already mentioned, we know more than 400 different types of restriction enzymes. In 1972, Stanley Cohen and Herbert Boyer. These are the pioneers who put efforts to create the recombinant DNA molecule, and this technology began the era of biotechnology industry. They laid the foundations of genetic engineering, and Herbert Boyer in 1976 made created a company called as Genentech, and in the United States, they for the first time this company ventured into creating the recombinant DNA molecules by recombinant DNA technology. In 78, somatostatin, the hormone, human growth hormone, the regulatory human growth hormone, was the first human protein to be made using this technology, that is the rDNA technology or recombinant DNA technology or molecular cloning. Now, if you see this technology, what is the basic which is needed? We say enzymes, okay, but with enzymes, when you get all the products, you need something where your gene of interest can sustain the processes which will be carried out till its final end product is achieved. Now, that requirement at the very basic level is a cloning vector. These cloning vector can be plasmids or phages. Now, today in this lecture, we are going to explore the various cloning vectors which are in the form of plasmids. So, what is a definition of a cloning vector? A cloning vector is defined as a small piece of circular DNA, preferably, or a system where a genetic material system where the DNA, which is the gene of interest, can be inserted and that will remain stable. That is cloning. Cloning vector. Cloning vector is a small piece of DNA into which a foreign DNA fragment, that is your gene of interest, can be inserted. There are various kinds of cloning vectors which are available in the field of research and the molecular biology technology today. The techniques which employ RDT or the recombinant DNA technology, these types of cloning vectors are plasmid vectors, as you can see, bacteriophage vectors, cosmid vectors. These three we will be seeing, and I will show some introductory light on the bacterial artificial chromosomes and yeast artificial chromosomes. There you can add a sixth point here, sixth type of cloning vectors, HACs or human artificial cloning chromosome, human artificial chromosomes, which is goes on as you go from plasmid, bacteriophage, cosmids. What is the hierarchy? The hierarchy from 1 to 6 is that as it goes on down the ladder, you can insert larger DNA fragments, that is a larger sized gene of interest, into your vectors. So this is the beauty of recombinant DNA technology. So as we are going to explore plasmids as cloning vectors, what are plasmids? Plasmids, these are extra chromosomal covalently closed circular entities which carry out those functions which are not present on the chromosome or which may be present on the chromosome, like a standby gene it has. A small example, if you want to take the lac gene or the lactose utilizing gene can be present on the chromosome, can be present on the plasmid. So as you can see here in the figure, this is the main bacterial chromosome and this is a small plasmid. This is the electron microscopic structure showing the larger original genetic material and these small circular plasmids which are scattered all around the cytoplasm. Now this plasmid has certain features, unique features. There can be a totally separate lecture of 1 hour on what these plasmids are, what are their types, etc., which you have studied in your third semester. So these plasmids, they are as good as the main chromosome, only they are limited in their potentials with respect to the size. As they are very small, comparatively very small, even if they carry out the function which is present on the gene of a chromosome, maybe or may not be, their kinetic potentials are limited. Means the rate of reaction is limited to a great amount. Means if a lac gene present on a bacterial chromosome has 10x capacities, then this plasmid has 1x capacities. Means 10 times the capacity of your protein synthesis is less, but nevertheless that cell attributes to that character, the cell possesses that character. Different types of plasmids are there. Plasmids which confer resistance to antibiotics, plasmids which confer resistance to heavy metals, plasmids which are responsible for structures like the pili formation, so on and so forth. These plasmids, they can replicate independently of the chromosomal DNA. When they replicate in two styles, that is a high copy number of plasmid and a low copy number of plasmid replication. A high copy number of plasmid replication is a cell which has a capacity to synthesize 10 to 100 numbers of the same plasmid when it replicates. Whereas low copy number of plasmid is the plasmid type which replicates whatever it may be. It achieves its copy number by replicating between 1 to 10 numbers. Okay, in all the cases these are usually double stranded and circular. Plasmids, they vary from over 1 kilobase to 1000 kilobase. 1 kilobase is 1000 nucleotides. A DNA fragment of 1000 nucleotides is called as 1 kilobase, and plasmids, they can vary in their size from 1 kilobase to 1000 kilobase, but the insert, that is the gene of insert which you are going to insert, that they are limited to 1 to 10 kilobase pairs. Only 1 kilobase pair to 10 kilobase pair DNA you can insert in a bacterial plasmid. That is the limitation. Now overall you have been seeing this process again and again, but again I don't get tired from showing you this protocol, which is for cloning or recombinant DNA technology or genetic engineering as such. Means simple steps, that is you first have a DNA, that is a gene of interest. The source DNA, you cut it into size, then take the gene of interest and insert it into a vector. Here I have shown you a plasmid, and you can see here this mismatch. What is this? Two plasmids joining each other. Now you see, think about all the probabilities which are happening. Plasmid, when it is open, you don't allow it to reneal. When you ligate it, two plasmids without the gene of interest. These are two plasmids. There should be a gene of interest here. There should be a gene of interest here. But instead of that, two plasmids, they circularize off. There is a probability. This is a mismatch. Now you see there is a second mismatch. The gene of interest. Instead, two gene of interest, they are end-to-end ligating. Now this two end-to-end ligated, there is also a mismatch. A perfect match is a plasmid which is entering or getting a gene of interest perfectly. Now to avoid all these mismatches, we have an array of enzymes functioning, which we saw in our last lecture. Various enzymes, DNA modifying enzymes. And what is the function of DNA modifying enzymes? To not allow such mismatches. Okay, so once you get a perfect recombinant DNA molecule, and that is made possible only with the help of a plasmid or another vector, any another vector, this has to be introduced into a host, and that is a bacterial cell which will have its original wild type characters, because as this bacterial chromosome is the recombinant DNA is inserted into the bacteria along with its chromosome, the plasmids will also multiply, the cells will divide, and one cell will form hundreds of cells. All these cells will get the plasmid with the gene of interest, and that is what is molecular cloning all about. So you see, now we are trying to explore the various characteristic features of this plasmid. What is the need, or in what type? What are the qualities desired in this plasmid? That we will try to explore. So this is, as you can see here in the figure, this is a plasmid, and it should have certain basic things like, for instance, the cDNA is the complementary DNA. It is also called as gene of interest, or the cDNA, or extra chromosomal gene which will express into your desired product. So naturally for this expression it requires a promoter. Most of the plasmids, they have the lac operon promoter because it can be easily induced by molecules of lactose. Then there is an antibiotic resistant gene needed. All these are artificially created. It is cut and paste technology. As you can see, restriction endonucleases and ligases, these are the important molecular tools for cutting and pasting. So a plasmid vector should have a promoter. Basically it should have a replication origin gene. It should have some detectable marker genes. I will explain what these are. And some sites. There should be restriction endonuclease cut introducing sites within it. And how many sites are needed? Many. So a polylinker type of system can be created. All this I will explain. So what are plasmid vectors? Plasmid vectors, these are defined as the carrier or vehicular molecules, DNA molecules which carry your gene of interest. These are the vehicles. Plasmid vectors are vehicles which carry the gene of interest. A vector, when combined with the gene of interest, this completely will be called as a recombinant DNA molecule. The aim of using this vector in the recombinant DNA technology is first to obtain multiple copies of the gene of interest which we have isolated, because this is our patent, this is our strength of the market. Whatever market you will generate or the industry which you will create, this gene of interest is your patented gene. So you need to obtain multiple copies, and to obtain this gene of interest in a desirable stable form, that is the product, to obtain the product from this gene of interest in a stable form, you need to express this into vectors. And these such type of vectors which not only maintain the gene of interest but allow the expression of such genes are called as expression vectors. So plasmid vectors are basically expression vectors. Now what are the ideal characteristics required in these vectors or plasmid vectors? Some 6 to 7 points I will mention. So first of all, it should be autonomously replicating unit. It should have some system like the gene of replication origins, which we have seen in DNA replication, which will allow the replication of the plasmid to multiple copies within the cell. And secondly, when the cell will, that is the host, will multiply, along with each binary fission and each cell division of the bacteria, whatever the plasmid copy number is maintained in this cell should be maintained in this cell. If it is, suppose let us say 78 is the plasmid copy number, a high copy number 78, when binary fission will be there, there will be a random distribution. Out of 70, maybe 70 are there and 8 have come over here. Listen carefully, listen very carefully. I am talking about the replication of this plasmid along with the cDNA. This is a bacterial cell. You introduce the plasmid with the gene of interest into the host. Now this will multiply 2 cells. Now this plasmid copy number with the gene of interest was having a high copy number of, let us say, 78 copies. Now when it cell division, binary fission is there, random distribution of this 78 plasmids takes place. 78 half and half, let us say, maybe 35 and remaining here, or maybe 70 here and 8 here, whatever, you know, or 18 here and 60 here, whatever. What happens after some time? This 60 will replicate until 78 is reached, and this 18 will replicate till 78 is reached. So in the end, when there is a completion of the process, both the cells will have that high copy number of plasmid number, that is 78. So this is the basic of autonomously replicating unit. So that the plasmid have first character, it should have autonomous replication system. Second is, this plasmid should be very easy to isolate and purify. Now plasmid isolation purification protocols are ancient, old, means old in the sense some 30-50 years they have been practiced, so it's very easy, just like the purification of DNA. But once it is purified, you should be able to check whether this plasmid is a mismatch or whether that is a clear recombinant molecule. Okay, usually the process which is used for introducing a plasmid containing your gene of interest into the host cell is a transformation technique. So transformation of the host with the vector should be easy. This plasmid is an extracellular DNA, and that will be uptaken by the bacterial cell, that will be introduced into the bacterial cell wall by transformation technique. This is artificial technique using calcium chloride technique, which I will explain in later lectures. So transformation is a process where this plasmid vector with the gene of interest is introduced into the host. So transformation of the host with the vector should be easy. Fourth point is that it should have suitable marker genes which will allow detection. What are markers? Now here you see the yellow colored segment here, it is a selection marker. So this is a resistant gene. What will happen if I introduce my gene of interest inside this marker? The resistance will be lost, cell will become sensitive to that antibiotic. Now if I am interested somewhere else, then this marker gene is intact. If it is for tetracycline resistance, then you can test it on plates containing tetracycline. In the end of this lecture, I will show you a process of how the marker, it plays a very important role. So right now, understand a marker is a gene which is responsible for some antibiotic resistance. The fifth point, characteristic feature, is it should have the ability to integrate either itself or the DNA insert which it carries into the genome of the host cell. That is, an integrative type of replication must be there, not abortive. Okay, abortive is it remains devoid of the chromosome, means both features it should have. If time is needed, means under stationary phase and starvation conditions, instead of getting degraded, it should get integrated into the host chromosome and sustain with the host chromosome. So the fifth characteristic feature of this plasmid to be used as a vector is it should have the ability to integrate either itself or the DNA insert it carries into the genome or the host cell. That is a very important feature. Sixth characteristic feature is it should contain unique target sites. What are target sites? Where the complementary DNA is inserted is a target site. Where the promoter will be used, that is a target site for a RNA polymerase. Markers are a target site for either insertion of a gene of interest or detection on the plating technique, whether it is resistant or not. So there should be genes which you have created. What is a marker in a plasmid or any recombinant DNA technology? Marker is a gene which we have inserted there for checking, testing, judging the validity of the process. Okay, whether the process is going correct or not. Time and again we have to keep on checking it. And how to check it? We check it with the use of markers. Markers are special appointed or specially inserted genes in the vector. Okay, so it should contain such unique markers, unique target sites. And finally, it should contain at least suitable control elements like promoter, operator, the ribosome binding sites, which will regulate the production of quantity, concentration of the RNA as well as the proteins which are synthesized using this complementary DNA. Okay, so seventhly, it should contain the basic controllable elements like promoter, operator, ribosome binding sites, then maybe the row dependent, row independent type of termination sequences which will result into a stem and loop type of configuration which regulate the protein synthesis, so on and so forth. These are the features which are needed. Now, general construction of vectors, what is the procedure which is followed? You can see here it is a simple plasmid, artificially created PBr 322 plasmid. This is just an example which is given in your curriculum. Hundreds of plasmids have been designed in the laboratory today and hundreds of different names have been given. But these are the pioneering, or you can say the historical invented vector molecules. So we use it as a yardstick to understand the modern vectors. So while constructing a plasmid vector, a DNA molecule, it should possess at least two essential characters. Means if this plasmid has to be used as a cloning vector, there should be two characters. One is it should have, it compulsorily it should have a gene of origin for replication, and it is the origin of replication gene which is required for autonomous replication of this vector plasmid using the host replication enzymes. Plasmid doesn't contain any enzymes for continuing with the replication. It is all dependent upon the host enzymes. Second important feature while constructing this vector is that it should have selectable markers which can be detected by plating techniques. Like you can see, two markers are there, one replication origin gene and ampicillin resistant gene and tetracycline resistant gene. Think very carefully, listen very carefully. These are selective markers or counter selective markers. What are selective markers or counter selective markers? For your understanding, remember these are two different antibiotic resistant genes. Both are antibiotic resistant genes. Now see, this is ampicillin resistant, this is tetracycline resistant. See the figure on your screen. Ampicillin resistance contains a restriction endonucleotide, one restriction endonucleotide where it can introduce a cut here. Tetracycline resistant gene has been designed which contains 1, 2, 3, at least 3 cut sequence. These are called as restriction endonucleolytic cleavage sites. It is the point here at the 651th base pair. The SAL1 restriction endonuclease will cut that DNA. Why it is necessary to cut this DNA? To insert your gene of interest. You see where else you can introduce the gene of interest. 1, 2, 3, 4, 5, 6, 7 different points are there where you can insert the gene of interest. Now if I insert it at 651 base, then it will be inserted within the tetracycline resistant gene and the resistance character will be lost. The cell will become tetracycline sensitive. If I introduce here, reverse will happen. Ampicillin sensitive, tetracycline will remain resistant. If I insert somewhere here, echo R1 here, it will remain both resistant because the genes are not disturbed. Okay, and you see very carefully we have taken a replication origin gene which does not contain the restriction endonucleolytic cleavage sites. Why the origin gene does not contain restriction endonuclease cleavage sites? Because it should remain intact for replicating the plasmid throughout the process. This is the basic mechanism for construction of vectors. There should be selectable markers. Ampicillin resistant marker is just one example. There are 4 different types of markers. Antibiotic resistant marker genes, herbicide tolerant marker genes HBR. This is used for agronomy and plant technology. Then metabolic or exotrophic marker genes, means like the maybe tryptophan operon genes, that is a metabolic or exotrophic gene. And screenable marker genes. There are genes which will, like the lac operon gene is a screenable marker gene, because if you introduce a lac and a lac mutant, you can have pink colonies and white colonies. You can have blue colonies and you can have white colonies in a dye detection agar. Okay, which is very popular in recombinant DNA technology. So screenable marker genes, there should be some gene which will be detected on the plating technique. So these are the general features for construction of a vector. Now, what are the different types of vectors? Classification of vectors on the basis of the aim of gene of interest. The classification is based upon 3 different perspectives. One is which is the type of your gene of interest, another is which type of host cells you are using, and third is what is the nature of the vector in the host cell. Nature of the vector's host cell means you have 2 different hosts. One is from where we are bringing the gene of interest, that DNA, that host, its nature. And here second is you are introducing this into a replicating host, its nature. So on the basis of nature of the vector's host and the nature of the inserting host, one gene of interest, host cells, there are different types of bacteria. On the basis of our aim of gene of interest, you have cloning vectors and you have expression vectors too. On the basis of the host cells which are employed, you can use bacteria, you can use yeast cells, you can use animal cells, you can use plant cells. So therefore, vectors for bacterial cells, vectors for yeast cells, vectors for animal cells and vectors for plant cells can be used. And on the basis of cellular nature of vectors host cell, prokaryotic vectors and eukaryotic vectors are present or are classified. Now let us see the type of plasmid vectors. What is a plasmid vector and what is its significance? So plasmids, which are the DNA, extra chromosomal DNA present in the bacterial cell, those plasmids, and phages which are parasites on bacteria, these are called as bacteriophages. Bacteriophage DNA is also very suitable vector, which we will see in our next lecture. So plasmid based vectors and bacteriophage based vectors are most common prokaryotic vectors. The prokaryotic vectors, they include plasmid derived vectors and bacteriophage derived vectors plus phagemid vectors, cosmid vectors, plain plasmid vectors and phosmid vectors. What are these? I will explain when I will explain the cosmid vectors in my future lectures. Today we are more interested in seeing a plasmid as a vector. So these plasmid vectors are the most common type of vectors used for the prokaryotic host cell. These plasmids, these are small circular double-stranded DNA, and they lack a, what you can say, covering or a protein or a coat or anything, and they exist as a natural extra chromosomal entity in the cytoplasm. They are as good as chromosomes, they are dwarfed by their metabolic capacities, they are very small in their size, and still they have the functioning capacities same as that some of the genes of the chromosome. So the plasmid as a vector, historically speaking, two plasmid vectors were designed which were very successful, PBR322 and the PUC series of vectors. We will be exploring PBR322 today as a part of your curriculum. The nomenclature I will explain, but PUC means a plasmid which was formed in the University of California. PUC is plasmid which was designed in the University of California, like that. So we will see the plasmid cloning vectors in the form of PBR322. Just a brief recap once again. The plasmid cloning vectors should have a replication origin gene. It should be able to make multiple copies of itself and express the gene which is inserted. Then it should contain certain marker genes, means selective and counter selective markers, preferably in the antibiotic resistance area. Okay, and it should have multiple cloning sites. Means as you can see here, this small segment of the DNA, it is capable of being digested, being cut by 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10 enzymes. So this is called as a multiple cloning site or a MCS. All these things are not available naturally. Remember, this is all artificial technique and we are designing as we want. So multiple cloning site is a design created by a genetic engineer. It doesn't exist naturally in nature. Why we are calling it an engineering? Means if you have a very posh bungalow designed by some engineer, that didn't naturally exist in nature. Okay, it was designed. Similarly, you have a design which is designed by a genetic engineer, and one of the features is it should contain multiple cloning sites. It is called as a polylinker. What is a polylinker? So the creation of a polylinker or a multiple cloning site is done by using a synthetic polylinker. So you have various types of nucleotides added, ligated, a double-stranded DNA is synthesized. How this modification of a natural DNA to a artificial DNA is done? Using all those enzymes which we studied in the DNA modifying enzymes. So you create a synthetic polylinker which has multiple restriction endonucleolytic cleavage sites, and you ligate it into a plasmid which is still, now this is not the gene of interest. Try to understand, we are introducing a synthetic polylinker into a plasmid vector. Now here comes the point where I say the limitation of the size. 1 to 10 kilobases only this plasmid can accommodate a gene of interest. Why? Because while designing this artificial plasmid, you have already inserted a lot of DNA. Okay, like this here, one of the examples. You take a small plasmid vector and introduce the polylinker. Now this has become a polylinker. I will go back and show you. So this has become a plasmid. Bit by bit, piece by piece, you introduce, then you introduce a promoter, you introduce a regulatory site, a ribosome binding site, and you design a plasmid, and it ultimately becomes of some size. And in the end, you need to introduce the gene of interest, which will have some size. So all this is limited. So the quest to introduce larger and larger DNA molecule in the vectors has resulted into different types of vectors like YAC, BAC and HACs. So this is how the polylinker is created. Now come, let us see the plasmid PBR322. This PBR322 is one of the pioneer plasmid to be created and used as a vector in recombinant DNA technology. Even though if it is absolute today, it is not in use today or very primitive for its use, It is known as the granddaddy of plasmids, and whoever who starts to study the plasmids starts with understanding PBr 322. Means there should not be a question about why this is such a old plasmid which is not in use, kept in for the curriculum or the syllabus. It is the granddaddy of all plasmids, the first plasmid to evolved, used successfully, and carried out many recombinant DNA technology procedures. So we will see this as an example of an ideal vector for recombinant DNA technology. The creation of PBr 322, this was done by Bolivar and Rodriguez, the two scientist friends on which this B and R stands. Plasmid, artificially created plasmid, which is small p, and when the vector starts with small p, you should understand it is a plasmid which is artificially designed in some laboratory. As I gave you the example, PUC was designed in University of California. So the scientists and public, they were concerned about the safety of recombinant DNA technology, and after the SLOMR2 and all recombinant DNA work which were done in the Boyer labs, they were under some doubts and confusion. In those time, two postdoctoral fellowships, Bolivar and Rodriguez, they engineered a safe and easy to use vector which was given a name on their initials, that is PBR, for B for Bolivar and R for Rodriguez, and 322 is the series. We will come to that. They constructed this plasmid PBR, and this PBR is a 4.36 kilobase plasmid. It has a replication origin gene. There are two genes which confer resistance to antibiotics. One is ampicillin resistant gene, the other is tetracycline resistant gene. This tetracycline resistant gene contains two restriction endonucleolytic cleavage sites, one which is from Bacillus amyloliqueficans, that is the BAM restriction endonuclease can act on this site. Other is from the SAL, that is SAL, which is from the same, which is within the same gene, that is the tetracycline resistant gene. There is a eco R1 on a plain site, means you can even introduce it without disturbing. You can introduce your gene of interest without disturbing the antibiotic markers. And there is one more antibiotic ampicillin resistant cleavage site present, which can be cleaved by restriction endonuclease by PST1. So there are several unique recognition sequences also. One is EcoR1, the other is PVU2, so on and so forth. It is a small vector made up of 4361 base pairs, which is around 4.36 kilobases. So this PBr 322, this was one of the first vectors to be developed in 1977. Once again, I repeat for your better understanding, the P indicates it is a plasmid vector. BR indicates designed by Bolivar and Rodriguez scientists, those who engineered this plasmid. 322 is the order or the indexing or the cataloging, that is, it distinguishes it from the other plasmids which were produced in the laboratories, which were labeled as 325, 327, 328, so on and so forth. Some cataloging or indexing was done by Bolivar and Rodriguez, that's their number. It is a 4361 to 23 base pairs, that is 4.3 kilobases, means it is less than 10 kb. It is a small plasmid vector. It carries two sets of antibiotic resistant genes, that is very unique, ampicillin and tetracycline. Tetracycline contains three sites for cleavage, where it can be introduced with the gene of interest at three different points. Ampicillin resistant marker contains one introductory site which can be cleaved by restriction endonuclease PST1. There are two sites apart from these restriction insertion. Plain DNA insertion can also take place by HIN3, EcoR1 or NDE at various points. So this is the construction of PBr 322. Now you can see each of the marker genes here in this engineered plasmid carries unique restriction sites for insertion of DNA into these sites. and if it is introduced into the antibiotic resistant genes, the genes are inactivated. Any one gene where the gene of interest is introduced, that will be inactivated. The new DNA, if let us say for it is introduced at PST1, ampicillin sensitive the cell will become, but it will remain tetracycline resistant, so on and so forth. If the gene of interest is introduced in tetracycline gene, the cell will become tetracycline sensitive but will remain ampicillin resistant. So here it is, tetracycline is called as a selective marker and ampicillin resistant is called as a counter selective marker. Means one is inactivated, it becomes sensitive. There will be no growth. As there is no growth, it is called as a counter selective marker. But the resistance means in the presence of antibiotic in the plate, the cells will show growth. They will show growth. So they are selective markers. So this plasmid PBr322 is a high copy number of plasmid. There are about 15 molecules present in transformed cells, but they can be amplified up to 1000 to 3000, means a artificially high copy number can be achieved, and that is what the cloning is all about. And in the presence of protein synthesis inhibitors, that is chloramphenicol, if you add, which inhibits certain reactions, only the plasmid can be amplified to such great extents. This vector PBr322, it comprises of a DNA derived from 3 different sources. Now the ampicillin resistant gene here, this is derived from R1 plasmid, and tetracycline resistant gene is derived from a plasmid which was artificially designed. It is called as a R65 plasmid. They are different plasmids, and origin gene from PMB1 plasmid. So all these are constructed, taken from different sources. Now that is what I want to show here on this collage paper. Once upon a time you were happy. This statement is taken from cutting and pasting alphabets from different newspapers. Maybe one is from Deccan Herald, one is from Times of India, one is from Indian Express, one is from the Hindu, so on and so forth. Different sources. Maybe one is from some magazine. Okay, the colored alphabets, maybe they are coming from some magazine. So we are cutting and pasting from different sources and we have created a sentence. We have created a collage. Similarly, these source genes are taken from different sources and they are joined, ligated to give you an artificial vector which is called as PBr322. So the construction of PBr322, it contains once again replication origin gene, selectable markers. It contains 2 antibiotic resistance genes which can be selective or counter selective. Okay, and within it contains cloning sites, the site where you will introduce a gene of interest. You have different options. You can introduce in ampicillin gene, you can introduce in tetracycline gene, you can introduce by using EcoRN without disturbing these 2 genes. Same with PVU2. So these are the cloning sites. Then use of PBr322, it is widely used as a cloning vector. It is widely used as a model system to study the prokaryotic transcription and translation. Means the prokaryotic transcription and translation can be regulated artificially in the laboratory by introducing the lag gene inside this. So this is also a very good model to understand the prokaryotic regulatory systems of protein expression. The advantages of this PBr322 is that it is very small in size, and due to the small size, easy purification and manipulation of this DNA is possible. Okay. Now 2 selectable markers, that is the ampicillin and tetracycline, these allow the selection of recombinant DNA technique by plating techniques. The recombinant DNA molecule, which we call as the chimera, or which we call it as cloned DNA, that when introduced inside a host, that host, whether it grows or not on a plate by some visually differentiating colony character, like a pink colony or a white colony or a blue colony or a white colony, that will define whether the gene of interest is introduced or not. Okay, so 2 selectable markers which allow easy selection of recombinant DNA technology by plating techniques. And one of the biggest advantage, beautiful facility which it provides is amplification 1000 to 3000 plasmids within a single cell. One single host can amplify, where by adding chloramphenicol you inactivate the host chromosome, chromosomal inactivity is achieved, and then you have a super amplification of the plasmid DNA. There are certain disadvantages of this PBr322, due to which now it is not being in used. But as I said, it is the granddaddy of all the plasmids, means it is this plasmid from where all the plasmids originated till date today. It has high mobility, means horizontal gene transfer, mobility is very fast, and there is limitation. The biggest thing, as we go further, you see, when you have started using mobile, you had less than 1 GB of RAM, you had some 100 MB of RAM, then you wanted 1 GB of RAM, 2 GB, 3 GB. Today mobiles are coming with 8 GB and 16 GB. Why there is a necessity for such advancement? And due to which the old mobile phones have become obsolete, they are not being used today. Similarly, limitation in the size of the genes which can be introduced, the gene of interest which has to be introduced is limited in this. Now with more and more options for recombinant DNA technology, we want larger genes to be inserted inside, and due to that, this disadvantage, it is not being in use today. But it is a best pioneered vector, initially originated, discovered, and on the basis of which all the most advanced vectors are designed today. So let us just run into the process how this PBr322 is used for molecular cloning. The technique of introducing a cloned PBr322 into a host cell is transformation, artificial transformation, which employs calcium chloride solution, and at 43 degrees centigrade a heat shock is used, and electroporation is a second technique. These two techniques we will be studying in the final part of our lecture series, that is the techniques for molecular cloning. I will deal it in with great detail, but here just remember two techniques are used as alternative, either transformation by calcium chloride or electroporation using high voltage. So basically the first step is PBr322 is cleaved by the ampicillin resistant element at PST. See here, at PST in the ampicillin resistant it is cleaved, the first cut is made, and in this cut the ampicillin resistant and tetracycline resistant, ampicillin resistant is chosen, and PST1 restriction endonucleolytic cut is made in the plasmid, as well as the same enzyme is used to obtain the foreign DNA or the gene of interest. This foreign DNA is cleaved and it is ligated into the PBr322. When ligation is successful and you have a good, that is not a mismatch, you have a correct recombinant DNA molecule, the tetracycline resistant element which is there, when ligation is successful, ampicillin resistance, if you are using, see here, if you are using the ampicillin resistance, this will be disrupted and tetracycline will remain intact. So we are using ampicillin resistance, so ampicillin resistance is disrupted and the tetracycline resistance remains intact. Okay, then this cells, why I am saying this is, now you have to detect whether your gene of interest has gone really inside or not. It is very easy. You take an antibiotic in the plate. In the next slide I will show you. But what is the next step? The next step is to transform this cloned DNA into the host cell. So E. coli are transformed and then grown on agar plates containing tetracycline. Now the plates are added with tetracycline. Why? Because it is tetracycline resistance has remained intact. If the cells are tetracycline resistant, they will only grow. So the E. coli cells are transformed on agar containing tetracycline, see here, and the selection of the transformed cells is done. All colonies they have plasmid, agar is containing tetracycline, individual colonies which are transformed to matching positions on the plate containing tetracycline and other containing tetracycline plus ampicillin. Two plates are taken, for one for selective marker, other for counter selective marker. From this plate all the colonies are transferred by replica plating. Means why we replica plate? Means you get to know the position of the colonies which are present there on the master plate. So these plates, all colonies with the plasmids on a plate which is containing tetracycline, means here what is the problem here? Can't you take all the colonies from this plate? No. Why? Because there will be plasmid in which DNA has not been introduced. That will also grow here. A non-recombinant only plasmid or a two plasmid circular to dual plasmid mismatch will also grow here. How to differentiate that from your recombinant DNA molecule? You make a replica plating of this on two plates. One is agar containing tetracycline, which is control, and agar containing tetracycline plus ampicillin. So here there will be different colonies, and cells which grow on tetracycline, listen very very carefully, the cells that grow on tetracycline but not with tetracycline plus ampicillin, which are growing here, these colonies are recombinant plasmids. So the cells which grow on tetracycline but not on tetracycline plus ampicillin containing recombinant plasmids, they have a disrupted ampicillin resistance, therefore the foreign DNA. So cells with PBR 322 without the foreign DNA, they retain the ampicillin resistance and they grow on both the plates. So this is how the identification of a cell containing the plasmid with the target gene is done. Finally, this is the real picture. Now whatever the picture I have been showing you here, this was for your understanding. Simple 2 to 3 genes I have shown. But come back to here, this is the genetic map of PBR 322. So you can see there are various linkers, there are various restriction endonucleolytic cleavage sites. Ideal plasmid will have as many as restriction endonucleotic cleavage sites as much possible. Why? Because that allows you to go for better options, more options. If not this, I will cut it at this point and check. Depending upon the size of the interest, depending upon the host, you don't know which type of condition may arise. So all the options should be ready. So originally the one which was created, you can see 4361 base pairs had so many restriction endonucleolytic cleavage sites, and that made it one of the unique vector plasmids. Finally, what is its state today? Today it is not being used because many other vectors have been constructed from its origin and more advanced facilities and provisions are available in the modern designed vectors. But still it is very popular commercial vector for studies for at the very basic level. There are hundreds of derivatives which can be designed from this. It is available in the market. It is taken, it is not used means it is not used as it is. It is modified, manipulated, more genes are introduced, more genes are removed. Some technological manipulations are done, but the base is PBR 322. Okay. So as Bolivar, the designer of this, what he says today, see, there is a very beautiful thing, very inspiring quote by Bolivar, scientist. He says that it is more meaningful to me that the original paper, that is the original plasmid which was designed, is not referred to anymore now, perhaps because of having achieved wide recognition. It is not referred to anymore in large number of papers. Why? Because now more advanced plasmids have come. So that is what Bolivar says. This was all with respect to the tools for molecular cloning with respect to plasmids. The next, equally competent, equally efficient, which can host a larger segment of your gene of interest, are phages. And one of the best example of phages to be used as the recombinant DNA technology cloning vehicle, the vehicle for taking your gene of interest and introducing in the host, are the lambda phages. So we will explore these lambda phages in our next lecture. Till that time, go through the links which I have given below. There are some animations which may help you. There are some sites which have beautifully explained theoretically in the form of notes. Refer those. Repeatedly see a couple of times my discussion and try to understand the beauty of molecular cloning process. I hope you understand. I hope you sustain the urge for a higher understanding. What happens next now should be the quest in your mind. For that, we will see next molecular cloning tool, phages. Till the time, good luck, goodbye.